Okay, good morning everybody. Thank you in particular to Victor for inviting me to be part of this conference today. And also I want to thank Victor because it was him who most helped me 7 years ago when we first started planning this program in 2003. We were at a meeting in the UK together when we started talking about the need to come and do some studies in the Chile Triple Junction area. And it took us 7 years later to actually get the first cruise. And as Victor just explained, after 7 years of waiting, we had a few setbacks. Working in the deep sea is always a challenge. On this particular cruise, we had the earthquake to contend with, also the tsunami, and then also we had an accident and lost our autonomous underwater vehicle as well on its 222nd dive, right near the start of the cruise. But as always with oceanographers, our team was very resilient, and what I'm going to present today are some important results we managed to get despite all of those setbacks, which I think have set the scene well for a lot more research in the future. The work we were doing as part of the CHESS program, so I just wanted, before going too far, to you've seen the Yeti crab earlier in Jesse's slide. I've been the co-chair of the CHESS program with Paul Tyler at the Southampton Oceanography Centre in the UK, and here's the team of all the other members that helped steer what our steering committee achieved. And so something I have to confess to you is I'm speaking here to the Census of Marine Life as a co-chair of a Census of Marine Life project, but I'm not a biologist. I'm actually a geologist and a chemist, but the reason I was involved in this program is because of my expertise in exploration using geological and chemical techniques to go out and study the kinds of, or to look for and locate the sites that biologists were most interested to study. We've been trying to understand over the decade what controls the biogeography and biodiversity of the kinds of ecosystems that are fueled by chemical energy at the seafloor, rather than relying on photosynthesis from sun and sunlight. And so we're particularly interested in hydrothermal vents and in cold seeps on the seafloor, but also whale carcasses and other kinds of environment that can basically produce chemically reduced compounds which can then interact with oxygen and other species in the water column to release energy that organisms can exploit for life. And so my role has been, as a geologist and a chemist, to go out and track down new vent sites in areas that people were interested in locating them, because you can't study the biology of a hydrothermal vent, for example, until you find the hydrothermal vent itself. This map actually shows the current state of the art. This is from our synthesis at the end of a decade of research that shows the different areas where we've found hydrothermal vents on the seafloor in the red circles, cold seeps along the ocean margins are shown in blue circles, and then the yellow areas, or yellow circles, show places where there are known whale falls on the seafloor that people have been able to conduct studies of. And you can see there's quite good coverage now, but also large gaps throughout large parts of the ocean basins. And so this actually rewinds and takes us back to where we were at the start of the decade. This dates from just after 2000, and this actually just shows in circles where the known hydrothermal vents were. In these circles you can see a very small section of the North Atlantic, some quite a good coverage down through the Pacific, down to about 30 degrees south, but not much further, and again good coverage in the West Pacific from New Zealand up off Japan. And the significance of the different colours is that all these different areas represent different biogeographic provinces. Different animals inhabited hydrothermal vents in all these different areas, even though the rock types are very similar and the chemistry of the fluids were very similar. And also, despite the fact that the entire deep ocean circulation occurs over the timescale of about 1,000 years. So the entire oceans mix on very short timescales compared to biological evolution. The habitats are apparently very similar in all these different areas, and yet somehow they've managed to sustain complete isolation genetically. And we don't understand why that should be, but it's very hard to understand why that should be when there are so many gaps and areas that we didn't understand. And so in our CHESS science program at the start of the decade, we identified certain areas of the mid-ocean ridge and also other areas of cold seeps and ocean margins that individual nations might be able to study individually, but we also came up with some very important areas that would require international collaboration. And the top two priorities that were there from the outset were, the first was to study across the Atlantic, that we knew that from the Gulf of Mexico to the Gulf of Guinea off Africa, there were cold seeps, or there should be, we would expect, there would be cold seeps in areas that are now famous for oil exploitation. There should be hydrothermal vents in the South Atlantic, although nobody had ever looked for them. There should be cold seeps off Barbados. There should be hydrothermal vents in the Cayman Rise. And again, there should be plenty of cold seeps that were already known in the Gulf of Mexico. And then the question was going to be, if we have all these different environments close to one another, how would their biology compare? Would the animals at cold seeps in the Gulf of Mexico be the same as cold seeps in the Gulf of Guinea, off Africa, for example? And would there be currents that would actually cause genetic communication between the two? And in the same way, would animals south of the Atlantic be the same as animals in the North Atlantic? And which of two different kinds might they be, given that we had two different biogeographic provinces very close to each other in the North Atlantic already? So that was one area, and many different nations got involved in that. The second area that I'd prioritised, which came up in our first meetings in 2003, was in this Chile Triple Junction area. And the reason for that is that the Chile Triple Junction is both geologically and biologically unique. It's the only place on Earth where a mid-ocean ridge, one of the driving forces of plate tectonics, is being subducted beneath an ocean margin through a subduction zone at the trench. There are trenches like this all around the rest of the Pacific Ocean. There are mid-ocean ridges around the middle of all the world's deep ocean basins, but this is the only place where the two intersect. So that's geologically very interesting. For example, the actual intersection is at an angle, and so through time, the triple junction, the point where the ridge is being subducted beneath the trench, has been migrating north along the Chilean border. Over the last 10 million years, it's extended up from Antarctica to reach at about 46 degrees south. To the north of here, we know that there are extensive gas hydrate deposits beneath the seafloor, and those things, as they become unstable and as they bubble out, drive cold seep communities that we know exist from Contribución and further north, and Javier Solanes will be talking a lot more about the biology of those systems later. To the south of here, as the ridge is subducted, it warms up the ocean margin, so probably it has driven off all the gas hydrates. There are probably no gas hydrates left to the south of here, but instead hydrothermal circulation is injected into the bottom of the sediment pile, and all the metal-rich fluids that normally escape into the oceans as particulate material instead are probably injected into the sediments and form very large copper and other metal-rich deposits all along the southern margin. So exploration to the south is probably somewhere where you'd find metal deposits. From the triple junction north is where you'd actually find gas deposits. So there's lots and lots of reasons geologically to be interested in this area. But what's really interesting for the Census of Marine Life and CHESS in particular is that what we should imagine is along this section of mid-ocean ridge, one might expect to find hydrothermal vents. Along here, one might expect to find cold seeps. And so this would be the only place in the world where one could study hydrothermal vent and cold seep communities in very close proximity. Now, the reason that's important is that some of the studies that had already happened and were beginning to be pulled together at the start of the Census of Marine Life decade showed that there was actually some overlap between animals that would inhabit cold seeps and animals that would inhabit hydrothermal vents. And there was also some overlap between animals that could take advantage and colonize whale carcasses on the seafloor as they fall to the seafloor and generate a large organic fall, which can then basically rot and generate a lot of hydrogen sulfide and methane release. And so there are animals that could exploit those systems as well. As we talked with Victor at that first planning meeting in 2003, we actually realized that the Chile Triple Junction area was actually even better poised for our purposes than we'd realized. Not only could we expect to find hydrothermal vents if we explored along these sections of mid-ocean ridge, we could also expect to find gas hydrates anywhere from the Triple Junction to the north, but also there was an expectation that we should be able to find whale falls because of the whale migration routes along here and the very particularly intense blue whale breeding grounds in the fjords in this area. We also have the oxygen minimum zone that extends all the way down the Pacific around about as far as Concepcion. And then also, of course, the large temperate forest, there would be large organic falls of different kinds continuing south. So basically, this is the only place on the entire planet where you could expect to find all the 5 different kinds of chemosynthetic ecosystem that we know about and study them together. So you couldn't ask for a better place to study biologically, and you couldn't really ask for a better place than Concepcion to actually have a laboratory as a headquarters to concentrate that study from. And if we can study all these different environments here, this could be like our Rosetta Stone for translation. That this would be the place that we could then go out, and this would allow us to provide better confidence for all our other interpretations about biogeography and distribution of animals from one chemosynthetic system to another throughout the rest of the planet. Because to argue that something can colonize a woodfall or a whale fall or a cold seep or a vent as it migrates from one ocean basin to another would require that they can actually make that kind of transition from one environment to another when they're all in the same place. So the Chile triple junction is the key place. And so our first cruise this year was actually to say, well, that's great in theory, but we actually need to go and prove that we do actually have these different ecosystems in close proximity, because until we've actually found them on the seafloor and done that first exploration, then we can't put in place our biological investigations. But a second thing that happened this year was the cruise took place in February and March, but I came straight to this cruise from a different cruise to the Antarctic, which was studying, as part of the same program, the first vent sites in the Antarctic. And what this shows you is, again, from the start of the Census of Marine Life Decade, some vent sites we knew should exist because we'd already done the exploration of the kind we've just been doing in Chile this spring. This kind of work was done 10 years ago. It took us 10 years to come back and visit the sites on the seafloor. And this shows the nearest known vent sites. This shows half the entire planet. So it's kind of a variation on the theme that Jesse spoke about before of how unstudied the southern hemisphere is, that at the start of the decade, there were no known vent sites anywhere in the South Atlantic. There were none known through most of the southern Indian Ocean, and again through the South Pacific. 37 degrees south was the end of the studies. This is where the famous Yeti crabs came from, but then there was nothing else known all the way around, so no venting were known there. That's not to say there shouldn't be any hydrothermal vents present. Over the last 20 years, anywhere we've gone and explored, we've never had to study for more than about 100 kilometers along any section of ridge before we found a new vent site. This speaks more to the lack of exploration that's been done in the southern hemisphere to date. And so what we were particularly interested in now with our discoveries was whether or not there was strong evidence for gene flow from any animals that might live at the Chile triple junction at vent sites here to vent sites in the Antarctic. And the reason we were interested in that is that some very new results that are currently in review from a British expedition that I was on in 2010 in January found stalked barnacles, these organisms here in the Antarctic, which are organisms that have previously only been found in the Southwest Pacific and also in the Central Indian Ridge. But the interesting thing is, these same animals are also things we've seen. This is just from an aerial photography or from overhead photography from an AUV from a cruise I was on with a Chinese ship in 2007, just south of Madagascar, to this section of mid-ridge here. So it looked like these animals and these animals may be very closely connected, which suggests there may be some South Pacific or Southern Ocean circulation which is homogenizing hydrothermal animals there. And if that proves true, then the interesting thing is that our Census of Marine Life program started with a very northern hemisphere prejudice, which was asking, why are the shrimp at vents at black smokers in the Atlantic different from tube worms at vents in the North Pacific? And maybe the question is, both of those are anomalies, and that in fact most of the animals in the Pacific, in the ocean at hydrothermal vents, are of some kind of Southern Ocean fauna. So that was one example of one species. And then the other one, which we've heard about as well, we know about the iconic Yeti crab, which when it was discovered on the southern East Pacific rise near Easter Island, was so new it basically defined an entire new family. What has happened since then, in the last few months, certainly in less than a year, is that the most abundant animal found at the hydrothermal vents on the East Scotia Ridge were also a different kind of Yeti crab, apparently. Now, that work's still underway, obviously, but this is a first result, is showing these things teeming in their hundreds all over the warm waters in Antarctica. But also, our chief scientist on the cruise to the Inspire region had also had a cruise earlier this year with Lisa Levin from Scripps to the Costa Rica margin to the north here, and they found another crab which they also believe is a different species of Yeti crab. So again, it suggests that there probably is some communication here between the East Pacific through into Antarctica, and again, so for a second reason now, the Chile triple junction becomes a really interesting place to go and explore, because it's sitting right on this communication pathway. So here are the results. The way we actually look for hydrothermal activity, you can think of a black smoker like a chimney, like in a factory, and it belches hot water and fluids and minerals and particles up into the water column, and then they disperse in a plume downstream. And so the first thing we do is we tow instruments through the water column, prospecting for the chemicals and for the particles in that plume to tell us whether or not we are within a few kilometers of a hydrothermal vent or if there is anything nearby. And so the first part of our expedition was to take an instrument package and just tow it all along the length of these two sections of the mid-ocean ridge. So the triple junction itself sits just here. This is the Chilean margin along here. This is the strike of the ridge here with a fracture zone offset, and then some more ridge section here. So we towed all along this section and all along this section, about 100 kilometers in total. So we've concluded that should be enough to guarantee we would find some hydrothermal signals somewhere. And the only place we actually found our signals was right at the southern end. I couldn't have asked for it to be a more perfect result if I could invent the hydrothermal activity myself. That as we came through the southern of the two segments, we were going through very clear water, so the blue scale here of particle enrichment is clean water. As we got further and further to the margin, the concentrations got higher and higher, a few hundred meters above the seafloor in this direction here. So it's in this general region along this first line, down through the middle here, that we saw particle enrichments. And so what we then did was we occupied a series of stations down to either side, where we didn't just use the in-situ sensors to know where there was smoky water, but to take water samples and to see if they had the right chemical enrichments, things like iron and manganese and methane and helium, to confirm whether this was hydrothermal or not. So this actually shows the results. In the numbers here show the different concentrations at the plume height in this particle-rich water for methane. And so you can see the highest concentrations are here and here. And what's really important is that along this axis then, there were actually two stations. The red trace was from here, which is out along the bare rock section of the mid-ocean ridge. But the black trace actually comes from here, right up against the tongue of right where the ridge disappears underneath the Chile margin, in a sediment-hosted environment. And then what was really important is that in between the two, we had a station here which had lower concentrations. So this actually means that this is almost like panning for gold. As you get stronger and stronger signals, then the signal gets weaker, and then it gets stronger again. It tells you when you've gone past more than one different source. And so this said that we had very high methane concentrations here, and also methane concentrations here that were high, but lower concentrations in between the two. So these were analyses we could do at sea that would give us a first indication that there was something in this area, but just the methane on its own wouldn't necessarily guarantee that this was high temperature hydrothermal activity. It could equally have been cold seep activity as well. So what we've done, one of the important measurements we've done at the same time since we've got back ashore, is to measure the helium isotopes in the same systems, and what we can see is exactly the same pattern. We can see the red trace here is from this station, and the black trace here is from this station, so these two are of equal magnitude. The fact that they're enriched in helium isotopes, the only way that helium comes from is from the mantle, so that requires that this is hydrothermal plume signal that we're seeing in both these two locations. And you can see the scale here. This is 5 miles, so about 8 kilometers. So these two are about 5 kilometers apart, one right at the triple junction, one actually up on the bare up part of the ridge, about 5 kilometers away. And again, we have intermediate concentrations here in the blue trace. Here is this profile in between, which shows that these aren't all part of the same source. There must be two separate hydrothermal features on the seafloor here. Now, ordinarily, the next stage of the cruise would have then been to launch our AUV and actually dive to this area and dive through the plume, map the plume out in more detail, this smoky water, go down and make detailed maps of the seafloor and take photographs. And that's the kind of thing we'd used previously on a German expedition in 2006 to the Mid-Atlantic Ridge, and the Chinese expedition in 2007 to the Southwest Indian Ridge. Unfortunately, this is where we had our third and final setback for the cruise, where we actually launched the AUV here. It went down. It was actually diving on the northern of the two sites. We were hoping it was going to come back with the first photographs of black smokers, and in fact it never came back at all. So we're pretty sure when we go back the next time we'll find the remains of the vehicle, and we know exactly what happened, that inside these pods there are glass spheres which provide the buoyancy for the vehicle, but they also host all the electronics. There are 9 of those spheres, and if any one of them ever implodes at depth, the energy it releases would actually trigger all the rest of the glass spheres. So we think we had all the different modes of electronics and communication on the vehicle all failed instantaneously at about 5 o'clock this morning, and the only way that would happen would be if all of those glass spheres had imploded simultaneously, and the amount of energy on that would have been about the same as 3 kilos of dynamite going off on the vehicle. So a pretty catastrophic failure. But hopefully there will still be enough parts of it that when we go back and find the vent sites in the future, we'll still be able to see the remains of Abe as well and see its last resting place. In the meantime, we didn't stop completely. As I said, we had our chemical results already. We also were able to use benthic trawls and sediment coring to conduct some preliminary biology. And what was really interesting was that this southern site, remember we sent the Abe vehicle to dive up here at one of the two sites. At the southern site, right at the triple junction, because there was sediment present, we were able to use a multi-corer to take cores, and what we found there was that the sediment that we collected was actually warmer than the bottom water. So again, it suggests we were in the right place. And then also, by using the sonar on the ship, driving along the ship's margin, along the continental margin, we were able to identify places where there were gas flares. We could see the bubbles rising from the seafloor, and when we conducted benthic trawling there, here are some of the animals we saw there. Here we found the tubes from Vestamintiferans, so we didn't find live tube worms, but we found recently dead ones, so we must have been very close on the verge of finding a cold seep. And also in here you can see we actually found a crab that looks suspiciously like a Yeti crab here as well, so a new Chilean Yeti crab potentially. So, and what's really interesting here is we know of the two vent sites, about 5 kilometers apart down here at the triple junction, but now we also have evidence for a cold seep site that we can go back and study as well, in much closer proximity to these two sites, but also one we can compare with the cold seeps known off Concepcion and further north as well. So although the Census of Marine Life is drawing to a close, an important thing to point out is that's not to say that international interest in this area has gone away. Most recently in June of this year, there was a meeting of the Interridge community back in Southampton, where Victor and I first started discussing this area in 2003. And you can see here are the top priorities for the international community for mid-ocean ridge research for the next decade. And the South Atlantic is important, the Central Indian Ridge gets mentioned, but the Chile Triple Junction remains a very high priority for international researchers. And that's not just from the Census of Marine Life perspective I discussed earlier, but also in terms of the formation of mineral deposits. A lot of geologists also are very interested in joining back in with research in Chile in the future. And how do we go about that? Well, I think there's a very real and practical way that we can actually imagine doing that in the future. I've also been in conversations with my colleague Colin Devi, who's the Deputy Director at the Institute for Marine Research in Geomar in Germany. They have a brand new deep diving AUV that can do a lot of the same work that Abe would have done this year. So they're very keen to come down here, and they have funding. He basically said anybody who's got a ship anytime, he's ready to come down here and start doing this work. The other thing I know for biologists in particular are very unsatisfied if all you can do is take photographs. I've learned this over the last decade for Census of Marine Life. If I go out with an AUV and take photographs of animals, biologists say, those are very nice photographs, but I really need the samples to actually study the taxonomy and to study DNA and do molecular techniques. And so what we've recently designed in Woods Hole is a very lightweight and very small and portable ROV that's battery powered and uses a single optical fiber. So again, it can be worked from very small ships. We've used it to dive to 5,000 meters from a ship as small as 40 meters. Now in Chile, what we actually have coming over the horizon is the brand new ship, the Cabo de Hornos, which is actually a 70-meter vessel. It's a very good design, excellent vehicle, and it's gone beyond being just a line drawing now and a theoretical thing to a real ship that's actually built. Again, it's had its setbacks with the tsunamis, but it's still going to be a great vessel for the future, and it has a long history and a long future. So I think we're very well positioned for a long period of international research that we could do together very easily using this combination of new technologies and a brand new ship that's going to be very well equipped for this purpose. That I think this is a great way to go forward for international research. And then the very last slide is then to extend beyond just the Chile triple junction, but also to talk about the longer term future for the next decade. That this is a repeat, a different projection of the same plot that Jesse showed earlier, which is one of the things that's coming from our synthesis, or our own synthesis from the CHESS program for the Census of Marine Life, but also a focus for a new program which is trying to pull together the different deep ocean realm programs. And one of the things we've highlighted is that the South Pacific, which is the largest single ocean basin, and hence the largest continuous ecosystem or habitat for life on the planet, is also our least studied. And so what we would like to propose is that there should be a plan in the decade ahead for a deep ocean realms to pull together for a program that can actually synthesize and study all the different aspects of the deep ocean realm across the breadth of the Pacific, including, not least, the Chile Triple Junction, as part of a much larger program. And so that is kicking off. There is a meeting next week in New Orleans in the United States to start talking about what the next synthesis should be. But once again, you can see with the areas that are of interest, then Chile is exceptionally well placed to play a leading role in that program. and a lot of international scientists are standing by ready to join in. So thank you very much.